All right, micro students. We are going to talk about chapter 4, the microscope and the gram stain. I know you're excited because we're going to be doing that for about 3 weeks. We're just going to have stain. So much stain. So we're going to talk about how to use your microscope because that's actually going to be a real thing. And I cannot get everyone's microscope, all by myself. So you have to lend a hand here. All right, let's talk about microscopes. Here is a delightful animation for you.
Light microscopes are useful for studying specimens ranging in size from about 1 millimeter to about 200 nanometers. It's impossible to study anything smaller by light microscopy because the shortest wavelength of visible light is about 390 nanometers, and anything smaller will not interact with the light. The most common form of light microscopy is called bright field microscopy. In this form of light microscopy, the light beam passes through the sample and into the objective lens. Let's look at the parts of a light microscope to see how a specimen is magnified and viewed. The lamp is where the light beam is initiated. This beam is divergent, spreading out as it approaches the condenser lens.
Okay, let's go back to the lamp. This lamp, there's a little knob on your lamp that makes it brighter or less bright, right? You need to fiddle with that if you can't see your sample. Step 1, fiddle with the light.
The condenser lens gathers the beam of light and focuses it so that it converges on the specimen with high intensity in a small area. The specimen usually is on a glass slide that rests on a platform called the stage. When the light beam hits the sample, the light rays are either reflected or refracted or absorbed by the sample. The beam then continues to diverge upward on its path away from the specimen. After the beam leaves the specimen, it passes through the objective lens. The objective lens performs the bulk of the magnification of the specimen. The eyepiece receives the light beam from the objective lens and refocuses it to recreate the image in the viewer's eye. Lenses found...
Okay, so you're going to mess with this, try and get it in focus. Then you're going to mess with the amount of light right here. Then you're going to mess with how condensed that light is. So how much focus is it going to be? Are you going to put all that light in a little tiny area or are you going to spread it out? So you're kind of going to fiddle with those 3 things. Okay, that is just to start us out. Okay, we did that. Why do we measure things in the metric? Because it just makes more sense. And honestly, our system makes 0 sense anyway. It's just simpler to do literally everything. General principles of using a microscope. What is the wavelength? What is the magnification? What is the resolution? And what is the contrast? You need to know the difference between contrast and resolution. It's kind of important. Don't worry, I'll tell you like 8 times. Essentially, also remember that your thing is upside down. It doesn't really come, honestly, microbes are usually like circular little blobs, so the top and the bottom don't really matter, but just so you know. It's helpful to keep that in mind. Okay, resolution. Resolution is the ability to distinguish 2 points that are close together. The better the resolution, the better 2 nearby objects are distinguished from one another. This is like pixels. This is, so you've got some blobbish, and you're like, I don't know if that's a rod or if that's just like a bunch of cones together. You need better resolution. You need to be able to distinguish those 2 things. Is it a big blob? Is it like a bunch of little blobs that are like connected to make a big blob? You think I'm kidding. I'm not. Here's just some things that you can see under our, we have the light microscope, right? So you're not looking at DNA. Nope, you're not seeing it. You're probably not seeing proteins. You're not seeing the ribosomes. You're not seeing any of that stuff, right? You're seeing typical bacteria. That's what we're looking at, okay? Okay, contrast. So we talked about resolution. A higher resolution is like a better picture, a sharper picture. You can distinguish things. Contrast. Difference in intensity between the 2 objects or between an object and its background, right? So if you play with your camera, you can like blur the background or not. That's contrast. How much does it differ from the background? It's important in determining the resolution, and staining increases the contrast. So if you have like a nearly clear bacteria, you're not going to be able to see it on our microscopes. So we stain it. That will be the lab that we do on Tuesday. You will stain it so that you can see what's going on. That increases the contrast between the clear light coming through and then the stained microbe. Again, use of light increases this. What is the difference between contrast and resolution? Anyone? Pause it. See if you can figure it out. Resolution is the sharpness, number of pixels in an image, the smallest detail that can be seen in the image. Can you read the person's little print on their shirt? The ability to distinguish between two closely spaced objects. Is it a bunch of little foci that look like a rod, or is it an actual rod? Can you distinguish what's going on? A higher resolution means a sharper image. All of a sudden it comes sharp. Oh, okay, now I can see it. Contrast is the difference between the light and the darkness. You can kind of see it, and you can see that it's different from something else, but it still is too light, too blurry up against the background. So that's the difference in the brightness or the color between the object and the background. The difference between the high contrast images have a wide range of color or grayscale. You can see where the stain, if there's more stain or less stain. And low contrast images feel dull. Okay? So again, how your microscope works. You're putting light here, then the light condenses it into do you want it in a tiny or do you want more light? Depends on how dark. We're going to do a negative stain, and that thing is super dark. So you're going to want all the light in all the places so that you can see it. If you have tiny little bacteria that are lightly stained, you're going to want to not have a ton of light on them or you aren't going to be able to see them. So that is all the light microscope. The light microscope, there's a series, right? You're going to start always on 10 and then go up to 100. And then at some point you're like, I still do not know what this is. I cannot see it. So what you're going to do is you're going to use some immersion oil. Yeah, you are. You're not going to want to because it makes a mess and it's hard, but you're going to end up having to do it. Why is that so much better? It just is amazing. You fiddle with it and you're like, oh my gosh, now I can see it. That's crazy. And that's because all of this light is focused right on there because you're not losing any. The immersion oil is actually absorbing that and pushing it through so that you're going to get way more light and it's going to be a sharper image. Does that make sense? That's why all of a sudden you're going to see it way better. You're like, oh my, look at that. Again, parts of the microscope, you're going to start fiddling here. You're going to start on 10. You're going to start 15. Then you're going to start here. You still can't see it. You're going to mess with the light. Do you need more or less light? More or less light. Then you're going to mess with the condenser. Maybe I need it condensed or more condensed. And you're going to just keep that up until you can finally focus it. And then you're going to yell for me. Again, big, here's your immersion oil. This is why you're not losing any of this light. It's going to be all right there. So what are we talking about? We are going to do a negative stain. You're really going to want to have a lot of scattered light, the maximum amount of light on that thing so that you can see it. The condenser, you're not going to want to have as much on that because you don't want a ton of light because it's already a light object. So some of you are going to be stellar with the microscope and some of you are going to be like, can you come focus this? Can you come focus this? Which is fine. I'll do it a bunch of times because it just takes, you just have to fiddle with it. And sometimes it takes me 20 minutes just to focus something. So don't feel bad. Here's microscopes that we don't have. So I'm just telling you, there are different kinds of microscopes. We don't have them. They're expensive. They're for very specific kinds of research. There's no reason for us to have them. Sometimes we do use fluorescence. Not in this lab, but it is possible to use fluorescence. And sometimes there are actual just fluorescent microscopes that look for things that fluoresce. We don't need a whole microscope to do that, but that is what it would look like if we did. Here's another one. There's confocal. Here's, again, the same light microscope, just telling you what to do. An electron microscope. These things are really amazing, really expensive, and no, we don't have one, and they magnify just cool stuff. Detailed views of bacteria. So if you're doing your unknown and you send me a picture that you took and it's clearly from an electron microscope, I'm going to know that you didn't take it. So, just be aware that that's the situation. Happens every year. Yeah, and some of them look like this. Did you do your unknown project on a transmission electron microscope? No, you didn't. So your image is not going to look like that. Just so you know. And it's you can see all kinds of cool stuff. Seriously, it's very cool. We just don't have them. You can use different probe microscopes. Here's all the kinds. I'm not super concerned about that. And I will see you in the next lecture. Oh, you can think about